Hey everyone, in this video I'm going to go through the lesson on glycolysis. Before we begin glycolysis, there's a couple of things I'm going to remind you all about and go over some basic bullet point conceptual items before we get into the actual pathway of glycolysis. First, a reminder of the equation of cellular respiration that you've probably saw in 9th grade, possibly even earlier. What we're going to do is take this big, broad equation and we're going to break it down into the separate biochemical pathways that are involved in the overall process of cell respiration. So we're going to take this equation and we're going to break it down into the actual subcomponents that are going on that contribute to this overall equation. The first pathway is glycolysis, which is the one we're going to cover in this video. The other two pathways are the oxidation of pyruvate and the Krebs cycle, which we will cover in a different class. In this video, we're going to focus on glycolysis, starting with an overall view or understanding of the process itself. At the beginning of this process, we start with glucose, which has 6 carbons. Next, we need to understand that glycolysis overall is an exergonic reaction, and that's going to involve catabolic enzymes. Remember that exergonic means that our delta G is going to be negative. So when I draw this graph, I'm going to put my reaction rate on the bottom, overall change in energy on the y-axis, and now instead of generic substrate, I'm going to write glucose as my starting product, or starting molecule. The ending molecule will be pyruvate, and we're going to see an overall change of delta G negative. With these exergonic reactions, don't forget, we can use that change in energy, the release of some of that energy, to phosphorylate ADP into ATP. Although some of the energy released is going to be lost as heat. Now, when we talk about the change in energy, remember that we're referring to the energy that is stored in the chemical bonds of the molecule. In other words, when you're looking at a carbon molecule with another carbon molecule, the energy is going to be stored in that covalent bond with those electrons. In this reaction, which is just a biochemical pathway, glucose is not only broken down, it's oxidized, meaning that it's going to lose electrons to a coenzyme carrier. Glycolysis occurs in all living cells, more specifically in the cytoplasm of all living cells. It also is an anaerobic process, meaning that it does not require oxygen. And it is the same 10 enzymes for all organisms. These last 3 bullet points are in a different color because we can use this as evidence that glycolysis was the method that Luca, or the last universal common ancestor, used this particular method. Now remember that not all cells have organelles, so the fact that this takes place in the cytoplasm is evidence for Luca. The fact that this doesn't need oxygen is also additional evidence for Luca because remember, on primitive Earth, there was no oxygen in the atmosphere. Oxygen did not come in until photosynthesis millions of years later. And the fact that it uses the same 10 enzymes for all organisms indicates that we all have common DNA because it's the DNA that codes for the proteins, and enzymes are proteins. Now we're going to take a look at the specifics of the biochemical pathway itself, that is glycolysis. And we're going to do this on the back of your page. We're going to start by drawing a basic scaffold that kind of outlines the progress of this biochemical pathway. We are not going to draw every single enzyme involved, and we're not going to draw all the products involved. We're just going to look at the big, broad overview. So this is what our basic scaffold looks like. And again, we're going to start with we're not going to go into details about each and every enzyme in this biochemical pathway, but you do need to know that there are enzymes controlling this chemical reaction. You also need to remember that in a biochemical pathway, the product of one enzyme becomes the substrate of the next enzyme. If you're confused about this, please make sure to go back and check on your flipped biochemical pathways we did earlier this unit. So we're going to go ahead and indicate what our starting molecule is, which is glucose, a 6-carbon molecule. And we're going to put our ending product, which is pyruvate. In fact, we end up with 2 pyruvates. Some textbooks will refer to pyruvate as pyruvic acid, so I've written it both ways, but they mean the same thing. Notice that pyruvate is a 3-carbon molecule, so we go from 1 6-carbon molecule to 2 3 carbons. So at the start of our reaction, we have 6 carbons, and at the end of the reaction, we still have 6 carbons, 3 plus 3. Now I'm going to take this overall biochemical pathway, and I'm going to divide it into 2 phases. The top portion of this reaction is called the energy investment phase. And as the name implies, we are going to invest some energy in the form of ATP so that at the end of the reaction, we end up with more energy than what we started with by incorporating or by breaking some of those bonds in that glucose. So we are going to invest 2 molecules of ATP into the top part or first part of our biochemical pathway. Remember that these biochemical pathways have enzymes and substrates and products. I've written a few examples of them as just E1, E2, E3. And the product of enzyme 1 becomes the substrate for enzyme 2. And then that product becomes the substrate for enzyme 3. Now we're not going to go into the details of the names of those enzymes or how many enzymes there are in the energy investment phase and the energy harvesting phase, but you do need to know that enzymes are there. So this ATP is going to phosphorylate one of those or 2 of those substrates, and what we're left with is ADP. And by phosphorylating the substrates, what you end up with at the end of the energy investment phase is a 6-carbon molecule, shown here in a very simplified version as just beads or circles of C's, carbons. We're not showing any of the hydrogen, we're not showing any of the oxygen, we're just showing a very simplified carbon chain. And this carbon chain has been phosphorylated twice by the ATP. The 2 phosphate groups are shown in yellow attached to this 6-carbon molecule. What's going to happen next is an enzyme is going to come in and break this bond right here between these two carbons. And what you're going to end up with is 2 3-carbon molecules. And so we're going to take this 3-carbon molecule and we're going to bring it over to this side of our reaction. And then this 3-carbon molecule, I'm going to go ahead and bring to this part right here. Once that 6-carbon molecule is broken into 2 3-carbon molecules, each with a phosphate group, we begin the next phase of our reaction, of our biochemical pathway, which is called the energy harvesting phase. In addition to generating or phosphorylating ADP into ATP, we're also going to see some oxidation of the 3-carbon molecule. Now remember, you can't have oxidation without reduction. So once that 3-carbon molecule loses its electrons, it's going to donate it to the coenzyme NAD+, which is reduced down to NADH. Now this happens for this molecule over here, but it's also going to happen for this molecule on this side as well. And so by oxidizing the substrate, NAD+ is reduced to NADH. We're also going to generate ATP by phosphorylating ADP. Now remember, this is occurring as a substrate whose name we don't know, but there's a substrate there is being converted into a product. And so as the substrate is being converted into a product, there is that release of energy. And we're going to capture some of that energy and use it to phosphorylate 2 molecules of ADP to 2 molecules of ATP. And it's going to happen on the other side of our biochemical pathway as well. And that process, as the ADP is being phosphorylated down up to ATP, as the substrate is being broken down, the capturing of that energy used to phosphorylate the ADP is called substrate level phosphorylation. So what I'm going to do is I'm going to take some of this additional information that we have learned in the overall process of this biochemical pathway and add it to the front of our page. This is a biochemical pathway for the breakdown of glucose, which is just one of cellular respiration. And so now to the front of my page, I'm going to add some of that information, starting with how much ATP is formed. So for total, ATP is made by substrate level phosphorylation. Sometimes the question will ask you, what is the net ATP? And if they ask you the net, you have to take into account how much we spent to start with. So your net ATP would be 2. We are also going to reduce 2 molecules, 2 coenzymes of NAD+ down to NADH. And in the upcoming days, we're going to talk about the fate of that pyruvate. What happens to that pyruvate depends on the organism and whether or not oxygen is present. Thank you all so much for watching. Bye.